My name is Justin. I'm an undergraduate student at Yale, and I've been working on a dimensionality reduction tool for exploratory analysis of the data in the exRNA atlas. I'll get started now. This will be a sort of an overview of some of the work I've been doing. It's in development, so any feedback you have or any questions you have, please feel free to ask at any time. So to begin, I'd like to do some acknowledgements. In case the presentation goes over, I don't want it to be cut off, and I'd like to thank all the wonderful people on this slide for their contributions. And there are, of course, many other people who have suggested great feedback, but these are some of the most significant. So thank you all. And let's get started. So what is dimensionality reduction? So it's the process of transforming data, usually in a tabular form or a table form, into a lower number of dimensions. So ideally, the low dimensional representation retains meaningful patterns or characteristics of the original data. And I'll show an example now, so maybe it will be a bit clearer. So this is an example of dimensionality reduction on a really simple data set. So you can see that on each column is a feature. And by feature, I mean it's a sort of variable, or we recorded something about each sample. And then we have the samples as rows. And I would say this is a data set with 10 dimensions, right? Because we recorded 10 features. And a really straightforward way to reduce this to one dimension would be to just take the first column, because you can see that it essentially represents all of the differences in the data set. You wouldn't say any data was being lost here, and you can actually completely reconstruct the original data from this reduced data set. Those are some properties that I think are useful in dimension reduction. But of course, very few real-life data sets can be so conventionally or conveniently reduced. And this is also, although this data set is not very relevant, I hope y'all can sort of start to see the applications of dimension reduction to a more complex data set. Because in theory, if we were able to capture a lot of the variance or a lot of the important characteristics in a fewer number of variables, that would be really useful in distinguishing, say, meaningful features or properties of something like extracellular RNA, which is the focus of this project. So I'll talk about a few approaches that I've encountered and that I implemented in this exploration tool. So first, principal component analysis, perhaps the most classic data set invented before the mid-1900s. So I also put the speed in terms of big O complexity. Essentially, you should expect smaller expressions in terms of n are better. So principal component analysis is the fastest of these methods. And it's pretty straightforward. You can imagine doing a best fit line is a type, sort of an analog, homologous to principal component analysis. You essentially repeatedly make best fit lines and express points in terms of projections on those lines to capture as much variance as possible. So I also listed the pros and cons of each of these methods, at least in the context of reducing data in the exRNA atlas. So the pros are principal component analysis is fast and straightforward, but the cons are it's linear and sometimes there's poor separation. We have t-SNE, another very popular technique. The speed is technically n squared if you naively implement it, but there are some optimizations you can make to approximate n log n. It's got very good separation and it's well known, but it doesn't scale well because, again, theoretically it should be O(n) squared, and the global structure is not very clear because distances between clusters are not very well illustrated. But I'll show some pictures later, so these become clearer. Then there are some more recent or sophisticated methods like UMAP, FATE, and a variational autoencoder, which is a deep neural network. And now let's get on to the actual exRNA atlas. I'm sure you're all aware of the objectives of exRNA Atlas and the ERCC as a whole. But I just want to thank again NIH Common Fund, ERCC, and the many contributors that generated the data I work with. These are some relevant citations. So now let's get to the actual visualization tool, which is under development. This is what the end user should see, and I hope that it appears relatively accessible. You have buttons, you don't need to do any coding, although there are some debugging features for more sophisticated use of the tool. Essentially, this first example would be, you see that there's principal component analysis being done on a data set of microRNAs with no subsetting of the features. So this is just sort of the simplest thing that can be done with the tool. You can see we are plotting the first component and the second component of PCA on this data set. And although the separation is not amazing, you can see some kind of clustering of various states or conditions of the samples, which are biosamples. So for example, you can see that there is the healthy subjects and healthy controls appear to be clustering in this blob. I hope you can see my cursor, but this is the overall workflow of the tool. So this workflow is not just to create the visualization, it's actually to create an app that creates the visualization. So the app itself is in this light purple lavender color. You don't need to understand all the details of this workflow, but I just want to give you an understanding of how you start with the raw data and you have an installer that installs this workflow, and then everything all the data is sort of processed in the gray area ahead of time, so that when the actual app is run and given to the end user, they experience the benefits of pre-computed, easily visualizable results without all the details in the middle. Additionally, this offers some protection in that most of the data will be completely anonymized after computation, so you don't have to release raw data in order to demonstrate the whole result. Although most of the slides I generated are from completely public data from the ERCC. So continuing, these are just some of the settings that are available for the tool. So you can see we have a bunch of drop-down menus on the left. You can make any combination of these work, essentially. So if you want to change the settings of the analysis, you just do so, and it will display the corresponding plot, usually within a couple seconds. So like if you wanted to scale all the data to be linear or logarithmic, if you wanted to choose the features with the top 100 highest variances instead of all the features, if you want to use some different normalization methods on the data before the dimensionality reduction, you can choose the method of dimensionality reduction, keeping in mind the nature of the data, and you can of course try different ones to see which one gives a good looking visualization. Once you've done the dimensionality reduction, I use a sort of second stage method. So you could look at the component, the 10 sort of components, you could summarize them, or you could apply t-SNE. The reason I use t-SNE as a second stage approach is because, again, as I mentioned earlier, it's a little difficult to scale, and most applications of t-SNE do some dimensionality reduction like PCA first. So this is sort of in alignment with common methods in the field. You could, of course, choose a percentage of features to subset, and there's the category, which is just essentially listing all the data sets in a grouped way. There's a few others. If you want to change what you color by in the plot, what you shape by in the plot, or what you filter by, these allow you to sort of exclude things you don't like or things you don't find are relevant. So for example, if you have unknown samples, you can exclude those and look at the plot in the absence of that. If you want to focus on a certain subset of conditions or sequencing methods, or even something like how long was the centrifugation of the RNA, you could choose to filter out certain things from each category. And of course, once you're done, you can see the plot, and you can also see the process data that generates that plot. You can download both of them on the spot and open them in anything you like, R, Excel, so forth. You can see the 10 principal components, each sample, and you can then see the corresponding metadata. So these are all the things you can label by, filter by, and so forth. And we have some instructions and some citations so that the tool is easy to use and understand and give attribution for. Those are, if we go back a little, those are the two buttons at the bottom. And of course, you can download those as well. I should also mention the tool can be deployed on a website, or it could be, you could download an offline version, or you could download a really lightweight source code version that connects to the online data sets, but it doesn't actually take all the storage. So another feature which I'll demo a bit, on an offline version of the application, is that you can bookmark things in the tool. So this is what the tool actually looks like. It's a work in progress, so I'm adding more data sets. I recently added fate, so you can see this interactive visualization under this dimensionality reduction method. And you can bookmark it. So if you were to use the actual tool online, it would have a really short statement after the URL, which would allow you to essentially replicate this exact configuration. So I think, yeah, so this is an overall look of what the tool would look like. You can change around the parameters however you want, and you can see that the updating is pretty quick. So then this is, for example, now instead of normalizing over the entire matrix, we normalize only over each column is normalized, each feature is normalized individually. And there are all kinds of other changes you can make. And of course, you can randomize the parameters as well. So back to the presentation, you can see I put the URL, which you can feel free to explore if you want. I'll paste it into the chat as well, but you can see it looks like this. And if I simply took one of the bookmarks, if I simply changed some setting and then I bookmarked it, you should expect that copy pasting in the bookmark would get you to the exact same plot. This should make it pretty accessible. So for example, if I just copy paste that link in and I go to the this, it should display the summary version of this plot. And if you give it a moment, I believe it should indeed load that in. Yeah, see the exact same plot. But back to the presentation, you can explore this on your own time if you wish. It's under development, so some data may not load. Here are some plots that I felt were useful and perhaps interesting, so feel free to ask any questions you want about them. These results are a little preliminary, but I do hope there are some interesting conclusions that can be drawn. This is just a straightforward principal component analysis, and you can see that you color by the condition, and the shapes represent the biofluid. This is a variational autoencoder, and comparing these two, you might say, well, hang on, this is a deep neural network. Isn't it supposed to be really good? You can see that some things separate out a little better, such as, say, this clump, colorectal and gastric cancer. Those separate out pretty well, but it does seem to be a bit more spaced out. And the reason is because, as documented in the instruction section, variational autoencoders try to have as even of a distribution over space. This is good for some data sets. For our data set, it's good because in theory, if I had person one had X level of gene expression and person two had Y level of microRNA being expressed, you could average those and it would still be a valid result. But if you average two pictures of cars together, it probably wouldn't look like a car. So that's just a good thing about the data we're working with that makes for it to make sense to have a variational autoencoder, which has this strength of being able to expect that points that are the average of 2 points should also be in the average of the location of the 2 points in the visualization. Maybe I didn't explain that very convincingly. So just to summarize, it's okay that the data is a bit spaced out and it's not perfectly separated, because once we apply t-SNE to the result, you can see some extremely good separation. And in fact, it seems to even be breaking conditions down into different subconditions, which I think it might be interesting. UMAP, as I mentioned earlier, is a pretty sophisticated method. It has a lot of mathematical foundation. And in this one, the local structure and the global structure both mean things. So that means, or have significance. So two clusters that are really far away are actually very dissimilar to each other. But as you can see in the TISNE one, the fact that, let's say if I annotate, the fact that this top cluster is super far away from this bottom cluster doesn't necessarily mean that they're so disparate from each other. They might actually be very similar because TISNE doesn't really preserve global structure, although the local structure makes a lot of sense in TISNE. But you can see for UMAP, the global structure actually puts all of these similar conditions together. FATE is another really interesting method for dimensionality reduction. Its potential of heat diffusion with, I believe, a temporal component. Allow me to, let's see, it should be, yeah, it's essentially assuming a temporal component. So that way you can, it's usually used for single cell sequencing, but in this case, we are using it on the data without assuming a temporal component exists. Nonetheless, it does seem to have some very interesting patterns in that it seems like all the conditions listed, such as subripian completion, all these pink conditions appear to occupy one common ancestry, so to speak. And many of the carcinomas are on another branch of differentiation. This is on rRNA, so it shows that the ribosomal RNA of various species found in biosamples can allow you to construct this. So you can make useful biological conclusions, not just from microRNAs or extracellular RNAs found endogenously in the body, but also in by comparing those the RNAs found to species that are not human. We also enable box plotting of various components. So for example, you can see that component 1 of this PCA on microRNAs appears to capture a lot of the variants in serum biofluids, stool, culture media, and so forth, but it doesn't appear to be capturing the variants in, say, cerebrospinal fluid. So this might this sort of look at each component may give an insight into what samples are contributing to that component being able to capture so much variance. We have summaries as well for plots for each of the methods. So for example, principal component analysis, the number of components should increase the amount of variance captured because we're essentially doing best fit lines over and over again. And you can see that 10 components usually captures a good portion of the variance. So that means we can reconstruct the original data, but it may not necessarily be meaningful nonlinear patterns. For the deep neural network, this is just a plot of the loss function. It's a little interesting because lower loss is better. So actually, you should interpret a lower number as a good thing. The testing loss is the loss on when we train the data, and the validation loss is when we do sort of intermediate tests in order to see how the training data is doing. So essentially, this plot shows that there is not overfitting because the testing loss is not going way below the validation loss. Overfitting would be if we fit some super complicated pattern, but when we tested it, it didn't actually recapitulate the pattern. For UMAP, a result recommended by the authors of the paper is to essentially use another clustering algorithm and see if the other clustering algorithm is better before or after UMAP. So before UMAP, clustering on the raw data appears to be pretty ineffective. You can see that there's a lot of noisy samples that the other clustering algorithm cannot appropriately group. But after UMAP, regardless of the number of the numbers after the UMAP here indicate the number of nearest neighbors, which is just a hyperparameter. That's not super important. If anything, this shows that the UMAP is pretty robust to the choice of hyperparameter for nearest neighbors. And in all cases, it allows the other algorithm to cluster a lot easier. So this indicates UMAP is pretty effective. I'll talk about another sort of not really a dimensionality reduction, but still included in the tool feature as well, which are sets. So upset is a technique that you can use to classify, or it's a technique you can use so that once you set thresholds for a gene being or a feature being present in a biosample, you can then sort of visualize how many samples possess that characteristic. This is essentially equivalent to a Venn diagram, but it's a little more sophisticated. So for example, you can see that colorectal cancer has 138 microRNAs that are uniquely expressed in that condition and not expressed in any other condition. And there are 89 microRNAs that are expressed in all of these conditions combined, except for systemic lupus erythematosus and so forth. The user is able to set what constitutes a gene being present, or what fraction of samples a microRNA needs to be present in for it to count. So that allows you to have a variety of profiles. Now, this is sort of a summary, actually. If we looked at the raw heat map, this is the plot that results. Now, on the bottom, it looks like there's a limited number of genes, but as you can see by the title, there's actually 2,500 microRNAs or features on the bottom. And each of these microRNAs constitutes a bar, and whether the amount of that microRNA is denoted by the color. So you can see there's a group of microRNAs that seem to be present in practically every tissue, and then there's a bunch that are more tissue specific, and some are barely present in any tissue, like the ones at the end. Another plot would be if we grouped, also these are sort of aggregated by a certain metadata type. So in this one, I grouped it by condition, but we can group it by anything you want, anything in any metadata label that's supported. So for example, like I said earlier, you could group it by whether it passes standards or the biofluid or anything you would like. You can see that there are some microRNAs that are exclusively expressed in female biosamples collected from female participants, and some exclusively expressed in biosamples collected from male participants, and so forth. And this is, you can also add a dendrogram. So you can sort of create these dendrograms, and now you see that the similar microRNAs are grouped in a way to make these sort of block-like structures. Yeah. And I believe there's one more by age, which I still need to verify. I'm not sure what the 100 to 110 is, but I believe it's some kind of issue with parsing the metadata. Other than that, though, age grouping may also produce interesting results. And that is all the slides I have for today's presentation. Any questions or feedback would be greatly appreciated.
Thanks a lot, Justin. I didn't want to call out the fact that you're an undergrad, but since you did, I just want to tell everybody this tool that you've developed is just amazing and really impressive. We've been very interested to find out whether releasing the URL to everybody in this workshop, the stress test will crash it or not. But if it doesn't, and in the days ahead, I think people should take this tool and try and find a new and interesting piece of biology in this ginormous data set. And if you find something interesting, take a screenshot and tweet it with the hashtag exRNA data 2021. We've got a fair number of questions for you, Justin. Let me just start. So someone asks, do PC1 and PC2, the principal components with the most that explain the most variability, do they have any clinical utility other than just dimensionality reduction?
That's a good question. So a good thing about principal component analysis is that you can add new samples to existing reductions. So for example, if you take a, this is just sort of speculation, but if I were to take a patient and sort of sequence their a biosample from them, and then I were to place it on an existing reduction, for example, if that patient were to appear with all the other carcinoma samples, that might be indicative. And that would be, of course, very far into the future, but potentially that would be a useful application in line with the ERCC's mission to develop useful diagnoses based on extracellular RNAs. But yeah, PCA is not the ideal, not the most sophisticated method we could use for this. Of course, experimenting with different methods is important too, and it would need to be supplemented with the context of sort of the patient's condition and so forth.
Another question is, can dimensionality reduction techniques be used to do differential gene expression analysis on RNA-seq or microarrays?
Differential gene expression, interesting. I would say it could give useful hypotheses to be tested by other means. You could, if you wanted to, you could look at a subset of useful genes and try to do dimensionality reduction. And if it captures almost all of the overall structure, that could tell you those genes are pretty important and they constitute the major features of your system. Although I think that's a really interesting, I think that's a really interesting idea you bring up, but I haven't done too much exploration in that direction.
Another question is, does the Explorer Tools workflow use all the data in the exRNA atlas, or can you filter it by how the experiment was performed, like using different RNA isolation techniques or sequencing methods?
Oh yeah, that's a great question. So if we go back a little bit, you can see that I've prepared for this demo version 2 data sets for each category. So for each of these categories, I either use all the data or I use the top 1,000 variables. I guess theoretically, we could take a subset of the microRNAs based on some condition. And you could take a subset of microRNAs and run dimensionality reduction on that, but it would take some time. The issue is that, right, subsetting the data before would change the result of the reduction, which is the bottleneck. But yeah, this tool has some features to allow you to subset the workflow allows you to subset the table, but it will not be instantaneous on the user's end. If like the user wanted to eliminate a sample from the analysis, that wouldn't be immediately feasible for them without waiting a bit at least.
Justin, go to the next slide. But he has done the dimensionality reduction on all the data, and you can filter with respect to the metadata. So if using all the data, the dimensionality reduction is done, and you could display the data sets for a subset of the experimental techniques.
Oh yeah,
maybe you filter on the metadata in terms of what you want to visualize.
Yeah, perhaps it will be useful to demonstrate it as well. Let me, oh, we can just do it right on here. There is a timeout to preserve server resources, but essentially, let's say I did not want to look at healthy controls. I only wanted to look at disease states. Then I could go to the filters and, okay, it seems that my bookmark left it on that. Let's say we tried UMAP and we did an exploration. Now I only want to look at I only want to look at disease states. I could eliminate all of the conditions that are, say, healthy control, like those 1,325 samples. And you will see that the plot no longer contains it in the sidebar. There's still healthy subject, which appears to be a different label. If I eliminate those as well, you can see the number of samples has decreased from 5,000 to about 2,700. And maybe this is more interesting to look at.
But just to the question, the original question, in addition to the conditions in terms of like the biological conditions, there's also the experimental metadata in terms of like RNA isolation kit and so on.
Oh yeah, those are also there as well. You could eliminate it in the final presentation, but the original reduction was done with the full data set. I hope that clears it up, maybe.
So we have one more question for Justin, but you may notice I did an experimental poll. Do people, please answer the poll. It's currently a statistical dead heat. Do people want to start answering questions now? Or do we want to break now if you want to get lunch or something and start at the scheduled time at 4 p.m.? Please take a look at that poll. And then Justin, the last question for you is, can you talk a bit more about the VAE architecture?
Oh yeah, sure. So this one currently has 3 hidden layers, and depending on the number of samples, it's essentially a geometric progression. So you can imagine this is your input, and then the variational autoencoder is trying to reconstruct an output, as you know. In the middle, we have 3 layers like this, and there's a few weights in the middle. And to actually do the reduction, we discard the right half and we only keep this part. And as you can see, I let it stay still for too long, so it just disconnected my connection. Yeah, that's a bit of an overview of the architecture. It's pretty similar to most autoencoders, but the variational a bit requires you to save not just the weights, but a distribution of probabilities in the middle. Yeah. Oh, and speaking of which, for example, if you did the offline version, you can see like the offline one, if I stop my share for a moment and reshare, is still running just fine. Yeah, I hope that answers the question.
Thanks, Justin. I guess the poll might be a bit confusing to people. We do have another session starting on the hour, but since this session has some missing speakers, my thought was we could answer some of the questions building up from earlier sessions. Let me show you something really quick. So I just want to mention a couple of other visualization tools that you guys should check out, and the developers of them were unable to speak here. So Anyuan Guo at the Huazhong University of Science and Technology has 2 tools for looking at RNAs isolated from extracellular vesicles, EV-miRNA and EV-Atlas. They're both at the website here. Their lab actually has quite a number of different tools. Like they have, for example, a tool for looking at RNA and platelets, and it's an interesting bioinformatics group. Their approach was to scrape all the data sets from GEO and SRA and do their own, RNA-seq processing and create their data exploration tool, which sort of fits with the ERCC's mandate of making all this data publicly available so the whole community can do whatever they want with it. So take a look at that. And then associated with the Human Biofluid RNA Atlas that Peter Mestach told you about is this R2 genomics analysis and visualization platform. developed by Jan Koster at the Academic Medical Center in Amsterdam. So there's the website you can look at to explore that.